microbiota. Please go ahead.
Thank you for the introduction, and thank you for the opportunity to be here talking about this topic. And just to let everyone aware, so this has been published in open access, so I will tell you I won't have time to talk in detail about all the information on this. I'm going to focus on some specific areas, but if you want to, you can have access to that, and I can direct you how to get that afterwards also. Okay, so my focus today is going to be about waste milk. We heard a little bit about that today at some different talks, but just briefly, so any time that we treat cows with microbials, very commonly we'll have, as a result, antibiotic residues in the milk of this animal. This is commonly called waste milk. Other factors that commonly result in the milk being called waste milk would be cows from having high somatic cell counts or some other condition that results in this milk not being suitable for consumption by human populations. So in the United States, we know that there's data that indicates that about approximately one in every 3 dairy farms feed pre-weaned calves waste milk. And that's actually, if you look at other countries and data available, we'll see actually very similar or actually sometimes higher percentage of the farms that actually feed this waste milk to calves. We have studies that indicate that feeding pasteurized waste milk compared to milk replacer to pre-weaned calves actually can result in financial advantages. So there's one study by Sandra Godden that showed about 70 cents per calf per day when feeding waste milk compared to milk replacer. However, one concern that has been risen and now more and more with concerns related to micro-resistance is what are the impacts of feeding this milk containing these drug residues to pre-weaned calves? More evidence of this concern is a recent report released by the European Food Safety Authority. They put together the panel on biological hazards, and as you can see, the title of that report is Risks for Developing AMR Due to Feeding Calves Milk Containing Residues on Antibiotics. So this is actually a very extensive report. They went through a lot of different studies that address this, and they talk about potential solutions, and if we are feeding this milk to calves, what are we doing with it? Where is it going, and how is it impacting the environment or other factors that could result in selection of a micro-resistance. So for this specific study, my aim was to try to evaluate what would be the impacts of feeding pre-weaned calves from birth to weaning milk containing antimicrobial residues on the functional microbiota of these calves. So for this study, in this study actually, I'll talk in the afternoon about another study that I have about waste milk, but this study we have a lot of different other projects that we based on this trial, but again today I'll only focus on the functional microbiota. But for this study, what we did, we collected newborn calves from one farm. We brought them into this enclosed environment where they had their own little individual stalls. Anyone entering those stalls or exiting, they had to use boots that were only to use on that stall, and they used protective equipment to avoid cross-contamination. These calves were housed in this environment for 6 weeks, and we had basically 2 different treatment groups that consisted of feeding calves whole waste milk. This milk was divided into 2 different groups. So the first group was called the calves that received drugs with no residues, or NR. The second group, we used the exact same milk. However, we added 4 different antimicrobials that are displayed on the screen, as well as their concentration. And the reason for selecting these drugs and their concentrations was actually based on a previous study that we conducted in upstate New York, where we collected waste milk samples from commercial dairy farms, and we looked at both at the concentration of drugs, first the presence, and then the concentration of these drugs in these waste milk samples using liquid chromatography mass spectrometry. And based on the average amount of concentration and most common drugs identified, we base this information on that. So these calves, again, were receiving this milk from birth to weaning, and this was commercial whole milk, so it was not pasteurized or not treated before that. And our whole objective was to try to have a very controlled environment that instead of conducting the study on a farm, we wanted to really identify what would be the effect of these drugs alone on the impact of the functional microbiota of calves. So for these calves, we collected fecal samples for shotgun sequencing at day 0, so at birth, 1 week of age, 3 weeks of age, and 6 weeks of age. We extracted DNA using power soil. After that, we used the Nextera XT for preparing the sample libraries for sequencing using the HiSec 2500. Following that, we processed the data, and mostly we used MG-RASP, and we used the seed subsystem to categorize the functions that we observed in this microbiota. And most analysis was conducted using JUMP and SAS. Okay, so this is a table that I'm showing, and again, the seed subsystem for functions, it's a hierarchical system. So first you'll have a higher category that could be cofactors, vitamins, prosthetics, and so on. And from that one, if you go within that category, you'll have other functions that are within that. And if you go furthermore in one function, you'll have a lower function. So it's a hierarchical, multi-level system. So what we did for this study, I used multiple linear regressions to look at what is the difference in the relative abundance of a specific function between treatment groups, between different weeks of age, and between the interaction of treatment group in week of age. And right here, I'm not displaying all the functions we found at this hierarchical level. We actually have 28 different functions. I'm just showing these 7 ones because these were the ones that at least we found a significant difference at the treatment group. And those are highlighted in darker black, yeah. For those functions, we observed that there were only 3 functions where we observed a significant difference between treatment group for at least one sampling time point. And that was for the ones that are in the yellow asterisk. So that would be nitrogen metabolism, regulation cell signaling, and stress response. So we further conducted took a honestly significant difference test to look at what specific sampling point did we observe a difference between the treatment group and the control group. From that, for all 3 functions, we observed that we only had a significant difference for samples S1, that would be samples collected 1 week after calves had been enrolled in the study. So we further conducted analysis within each one of these 3 functions at lower levels. From that analysis, and again, here I'm starting with the regulation and cell signaling, we observed that it wasn't consistent that we only saw a greater relative abundance for a function on one treatment group versus the other. As science, in biology, it always makes it a little bit more confusing. So we saw different trends and different functions. So for example, for the calves receiving drug residues in the milk, so the red group bars, we saw that were a higher relative abundance of genes related with zinc-regulated enzymes, with the PGPP metabolism, and with the staphylococcal accessory gene regulation system. In these, for example, the zinc regulation enzyme could provide some advantages for that microbiota to adapt to different levels of zinc in the feed and could make that microbiota more robust. Unfortunately, I won't have time to go through all of them, so I'll try to give at least an overview of what we observed. And the 2 other genes functions we observed, the PPGPP and the SAR gene, usually have been related with microbiota or bacteria exposed to stress, and we'll usually see that more commonly. On the other hand, for calves that receive no residues in the milk, we observe a higher proportion of the orphan-regulated proteins, which are proteins that we don't know exactly their function. That's why we call them orphan. And we also observed 3 other functions, the DNA binding regulatory protein, the TYRR and the CYTR. And those are actually genes that are related with ability for the microbiota bacteria to conduct transcription and adapt to different stresses also. For the nitrogen metabolism, we observed that there was a significantly higher relative abundance of genes related with ammonia assimilation on calves receiving drug residues in the milk. And we know that ammonia is the preferred source of nitrogen for bacteria. So again, that could indicate that this microbiota would do better with higher availability of nitrogen. Interesting enough, for the calves that did not receive drug residue in the milk, the blue bars, we observed that they had a higher relative abundance for these other three functions that are actually related to trying to use different sources of nitrogen other than ammonia when available. So this could be just the ability of this microbiota to adapt easier in an environment where ammonia may not be available in the diet. For the last category, stress response, for the calves that received drug residue in the milk, we observed a higher relative abundance of the heat shock DNAK genes cluster. And these genes have been linked to exposure of bacteria to stress. It could be antimicrobial, could be changes in pH or other stress factors. And it's one way that bacteria adapts to these changes and tries to protect the bacterial DNA from the stresses affecting it and resulting in death. For the calves receiving no drug residue in the milk, we observed actually these three different functions that are all kind of linked together as they relate to ability of the bacteria to osmoregulate. And for calves, usually for me, I would see this as being important if they're feeding milk replacer and whoever's feeding the milk didn't really adjust or didn't mix the milk right, and you have this different inosmotic pressure. So maybe they would be able to more robustly adapt to this environment and survive. Okay, so overall, for conclusions, we really just observe a significant difference for function at 1 week after we expose these calves to this treatment. Again, for week 3 and week 6, we did not observe a significant difference. So it could be, as we have observed with other studies, that we looked only at the composition of microbiota, that this microbiota is more vulnerable in the beginning, and these antimicrobials are playing a bigger role. But again, this is just some initial information that provides some more clarity that, okay, antimicrobials, even at these low concentrations, can affect the microbiota. We know that some specific functions that it affects, but again, what does that translate into reality to the producer, and what can we translate that? There's still a lot to learn on that point. I just want to thank all the farmers that collaborated with this project too, and my team. And if there's time for questions.
Okay, thank you very much for your interesting presentation.